Thank you. So I thank the organizer to be able to present this work that we recently published in Journal of Immunology. So I will talk about the B-cell response, and the role of the B-cell and antibodies in HCV infection is still currently debated. So it is generally known that the antibody response is slow and delayed during acute infection. However, antibody can be neutralizing both in vivo, in vitro and in vivo. Those antibodies can induce escape mutation from the virus, but the generation of an early antibody response was associated with virus clearance in several studies. There are other studies who showed spontaneous resolution without seroconversion, so in some cases you don't seem to need the antibodies. And in the context of reinfection, the generation of cross-reactive antibody was observed. So our goal is to really better define the role of the B-cell and antibodies during HCV infection. But in order to do that, we need to be able to identify HCV-specific B-cells. And for that, we developed a fluorescent B-cell tetramer based on a viral protein. So we used the E2 glycoprotein, the surface protein, and a construct that was used to purify the protein and determine the crystal structure. So this construct was shown to produce good amount of properly folded E2 protein. And we've used it with a biotinylation site and protein A tag to be able to purify the protein. And so the biotin will enable to interact with streptavidin, so incubating our antigen with streptavidin will enable to form the tetramer, so having 4 liaison sites. And then the HCV-specific B-cell will bind to the tetramer by their B-cell receptor, and having a tetramer instead of a monomer will enable more receptor to link to the tetramer and have a better detection, which can be done by flow cytometry, and you can also isolate the cell for downstream experiments. So first, we validated that our tetramer can bind E2-specific antibodies. So we use antibody that was coated on beads. And you can see in white in the first graph that our E2 tetramer can bind to E2 antibody, but not to HIV antibodies. And then HIV tetramer can bind to HIV antibody, but not to HCV antibodies. And the second validation we did was with hybridoma cell line, and we can see those hybridoma will produce either HCV antibody or HIV antibody, and our tetramer binds only to the HCV antibody-producing hybridoma. So we show the specificity of our tetramer. Then we wanted to see if we can detect HCV-specific B-cells in infected individuals. So we work with the Montreal Acute Cohort Study, which recruit people who inject drugs that are HCV naive, and we follow them regularly and take blood samples until we get to a primary infection, which is detected either by RNA-positive test or an antibody-positive test. And we follow them closely during their acute infection until they either spontaneously resolve the infection or develop a chronic infection. So first, we work with chronically infected patients to try to identify those HCV-specific B cells. And we use flow cytometry, and we focus mainly on class-switch memory B cells. So those memory B cells will express CD27. They will have class-switch, so they will then be negative for IgM, and they will express CD19, and they will bind to our tetramer here. So first, we use a control group, which was LT donors and HCV-naive PWIDs. And in those people, we could see barely no binding to the tetramer, and we use this control group to establish our threshold of binding. And then we use chronically infected sample from chronically infected patients from different HCV genotype. And for all the genotype, we could identify HCV-specific B-cell population in most of the samples. Then we wanted to see if we could see those cells before the establishment of the chronic infection, more in the acute phase. And so we used 3 different time points, so people that were followed longitudinally at a baseline time point before the infection, at a late acute time point around 5 months post the infection, and in the chronic phase. And even though we could see an HCV-specific B-cell population at the chronic phase for all the patients, the detection was limited in the acute infection, suggesting that the B-cell response is indeed delayed in those patients. Then we wanted to look in more detail into those Hcv-specific B cells to try to identify some antigen-specific characteristics. So we sequenced the B cell receptor from those cells, and we were looking for a focus repertoire, so suggesting that some cells were really selected, and accumulation of mutation, which happens in the maturation of the B cell response. So we sorted Hcv-specific memory B cells and also naive B cells as a control. And so first we look at the repertoire, so all the different sequences from that are found. And if we look in the naive B cell first as a control, we could see that there is a very broad repertoire, which was expected. So we could find thousands of different clonotypes, which were all at a very low abundance. But when we look at the Hcv-specific population, we can see that it's really different. So we could find only just a few clonotypes, only a few different cells that were able to bind HCV protein, and they were all almost all in a dominant population. So this suggests that those clonotypes were indeed selected and amplified during the immune response. And we also observe an increase in the mutation frequency of those cells, suggesting that the cells pass through the affinity maturation process. So with this, we showed that this new tool can successfully identify HCV-specific B cells from different HCV genotypes. And we could identify class-switch memory B cells specific to HCV in most chronically infected patients. However, the detection was limited in acute infection, which would suggest either a delayed response or a response of too low affinity to bind to our tetramer. And finally, we could sort those HCV-specific B cells to analyze their BCR repertoire, and we showed that their repertoire was focused, so these cells were selected and amplified, and they had an increased mutation frequency, suggesting the maturation process. We will now be doing the same work, but comparing different groups. So we are really interested to compare those who spontaneously resolved the infection with those that develop a chronic infection to see if their B cell response is different, to try to find some correlates of a protective immune response, and also look in the context of reinfection. We will also look at the BCR repertoire in those different groups. And finally, it will be really interesting to establish correlation between the B cell response, but also with the CD4 follicular helper cells that are the CD4 cells specialized to help the B cell response, and also establish correlation with the generation of neutralizing antibodies. With that, I would like to thank everyone in the Shukri lab and also our collaborator, especially Dr. Arash Grakwi and his team who developed the Tetramer, and also our cohort, and I thank you for your attention.
We have time for a couple of questions. So, is there any thought to expanding your cohort to co-infected patients, Hep C and HIV co-infected?
In our cohort, they are not present, so they are excluded from the cohort, but I think it would be something really interesting to look, maybe to do some collaboration with other cohorts to see if they have those cells.
Very nice presentation and really very original concept. I have a question following up on what you said at the end regarding neutralizing antibodies. Did you look already preliminary in these patients whether there are differences, whether they are in cross neutralization, for example, and strength and breadth of the antibodies?
Yes, this is a really interesting point, and it's something that we're starting to do now, again in collaboration with the lab of Dr. Grakwi. So maybe next year I can talk to you more about that, but we don't have the results yet.
Great. Thank you very much.
Thank you.